Imagine Earth as a stage dressing, slowly shifting its backdrop, glaciers retreating like curtains pulled aside, seas warming, and a world holding its breath before a new kind of life steps into the light. This is the Ediacaran, a distant chapter in Earth's deep history, when life first unfurled beyond single cells into curious, often alien-looking multicellular assemblies. It is a story of slow planetary change and sudden biological invention, a place where strange shapes carpeted shallow seas and where the rules of life on Earth began to feel more familiar to the forms that would later dominate. The Ediacaran lasted for nearly 100 million years, from about 635 to 538.8 million years ago. To put that into perspective, it began after the Cryogenian Period, an age when Earth was so cold that glaciers may have reached the equator, the infamous Snowball Earth, and it ended just before the Cambrian, the era when familiar animal groups exploded into diversity. The Ediacaran sits at the hinge between these worlds, the final period of the Proterozoic Eon and the last breath of the so-called Precambrian Superion. It is a transitional zone, a slow dawn at the edge of a biological day that would blaze into the Cambrian Explosion. Our story gets its name from a sun-baked ridge in South Australia, the Ediacara Hills. In 1946, a geologist named Reg Sprigg stumbled upon impressions in the sandstone there, impressions that did not look like anything paleontologists had seen before. These weren't shells or bones. They were soft-bodied impressions, fronds, discs, and quilted ovals that hinted at whole, unfamiliar kinds of organisms. Those impressions became the first glimpses of what we now call the Ediacaran Biota. And for decades, scientists debated what these creatures were: animals, giant algae, lichens, or representatives of a failed experiment in multicellularity. In 2004, the International Union of Geological Sciences made it official. The Ediacaran became a formal geological period, the first new period recognized in 120 years. The type section, the geological golden spike that marks the official base of the Ediacaran, sits not in the Ediacara Hills themselves, but about 50 kilometers southeast in the Inorama Creek bed at Brachina Gorge. There, a particular carbonate layer, a cap carbonate, caps the glacial deposits left from the Marinoan glaciation. That cap rock shows a distinctive chemical fingerprint, an abrupt depletion in a heavy form of carbon, and it serves as the signal that the deep freeze had ended. It's a kind of planetary exhale recorded in stone, marking the transition from global glaciation to a climate where complex ecosystems could take hold. What happened after that exhale was remarkable. The seas became a canvas for living architectures not yet seen. Soft-bodied organisms that left impressions of fronds like underwater ferns, quilted mattresses that suggest internal compartments, radial discs, and bizarre three-rayed shapes. Scientists have given names to groups of these life forms: Proarticulata, with segmented bilateral forms like Dickinsonia and Spriggina; Petaloname, waving frond-like bodies such as Charnia; Aspidella, radial discs that were once misunderstood as inorganic; and Triloboszoa, with triradial symmetry like Tribrachidium. Some of these forms are so unlike modern animals that they invite metaphors: quilted pancakes, feathered fans, concentric ripples. Around 575 million years ago, a relatively sudden radiation of these morphologies occurred, an event sometimes called the Avalon Explosion. It wasn't an explosion of thousands of species overnight, but rather a notable increase in the diversity and scale of multicellular life. Many creatures of the Avalon fauna are soft-bodied and delicate, preserved only when specific conditions allowed rapid burial, in microbial mats or fine sediments, so their outlines could be fossilized. These fossils have been found in places as far-flung as the White Sea region of Russia, Newfoundland, Namibia, and Australia, painting a picture of a global, if still patchy, ecosystem. Yet not everything from the Ediacaran resembles the animals we recognize today. Some fossils are enigmatic, occupying a liminal space between what we call animals, algae, or even unique extinct experiments in body plans. Others, however, are more readily allied to modern groups. For instance, fossils such as Aurora lumina and Hauudia suggest early cnidarian-like animals, relatives of jellyfish and sea anemones, while Yellingia hints at motile bilaterians, creatures with a front and back and the potential for directional movement. Kimberella shows mollusk-like features, and sponges, the simplest of animals, appear in the fossil record by the terminal Ediacaran. These holdfasts, fronds, and disc-shaped impressions form a record of life learning to build bigger, more complex bodies with differentiated tissues. If you look at the Ediacaran Biota chronologically, four broad phases emerge. The first, spanning roughly from the beginning of the period to around 575 million years ago, is dominated by large, ornamented microfossils, acritarchs, tiny organic walled forms that suggest planktonic or algal life was flourishing. The second phase, the Avalon interval, brings those quilted and frond-like forms into prominence. The third, known as the White Sea Biota, about 560 to 550 million years ago, is named for an area where these fossils are abundant and diverse. The final phase, the Nama assemblage, lasts from roughly 550 to 539 million years ago and includes some of the last lasting impressions of these ancient communities, alongside the earliest appearances of mineralized shells. The ending of the Ediacaran is as intriguing as its middle. Many of the iconic Ediacaran forms vanish before the dawn of the Cambrian. This disappearance has stimulated vigorous debates. Was it a mass extinction caused by newly evolving animals that altered the seafloor environment, an anoxic event cutting off oxygen to coastal ecosystems, or a combination of environmental and biological pressures? One idea is that the appearance of burrowing animals, creatures that churned and oxygenated sediments, changed the structure of microbial mats and the shallow seafloor ecology, making conditions less hospitable for the stationary, soft-bodied Ediacaran residents. In another view, widespread drops in oxygen or chemical shifts in the oceans could have imposed stresses they could not survive. The truth may well be a complex blend of both ecological innovation and environmental upheaval. Geology and ice tell another layer of the story. The Ediacaran saw the supercontinent Pannotia form and then begin to break apart, reshaping ocean basins and coastlines. Several glacial events punctuated the period, such as the Gaskier's glaciation and the Bacanurian events, though the causes and global significance of these glaciations remain areas of active research. A significant chemical event during the period, known as the Charnia excursion, produced a striking negative carbon isotope signal in carbonate rocks. At first glance, large negative excursions in carbon isotopes were often linked to glacial happenings, but the Charnia appears disconnected from ice records and lasted millions of years, hinting at complex ocean chemistry shifts rather than simple freeze-thaw cycles. Dating the Ediacaran posed another challenge. The very place that gave the period its name does not have easy igneous layers to date directly with radiometric methods. Instead, scientists stitched together a global quilt of dates from far-flung sections, Africa, China, North America, Tasmania, using uranium-lead and rhenium-osmium chronometers. Those cross-continental correlations align the start around 635 million years ago and the end at about 538.8 million years ago, a span of roughly 96 million years. Geology is in part an exercise in detective work. Find the clues in rocks, compare the chemistry, and tie timelines together. One interesting nuance of Ediacaran stratigraphy is that its beginning isn't anchored by the appearance of a new fossil type, unlike many other periods where a recognizable organism marks a boundary. Instead, the lower boundary is set at that cap carbonate layer, a chemical signal of a global transition out of the Marinoan glaciation. The upper boundary, meanwhile, aligns with the base of the Cambrian, associated with trace fossils of burrowing animals like Treptichnus pedum. Using chemical markers and trace fossils to define boundaries comes with debate. Cap carbonates aren't ubiquitous. Isotope signals can vary regionally or be altered over time. And trace fossils like Treptichnus have appeared below their supposed horizon in several places. Yet these choices reflect the best integration of geochemistry, sedimentology, and paleontology available to tie deep time into a shared global chronology. The Ediacaran seas were not static backdrops, but dynamic arenas where life and environment interacted. Microbial mats, carpets of bacteria and microbes, were common on the seafloor. These mats likely stabilized sediments and provided a substrate for many Ediacaran organisms to live upon or anchor into. They also help preserve fossils by trapping fine sediments and creating low oxygen conditions beneath that slowed decay. The presence of microbial communities suggests landscapes very different from modern sandy or muddy seafloors, and their decline, whether due to grazing, bioturbation, or chemical change, may have been pivotal in reshaping ecosystems. Even the moon played a role in this ancient theater. Back then, the moon was closer, so tides were stronger and faster. Days were shorter, about 21.9 hours long, and a year contained more solar days than today, with estimates suggesting around 400 solar days per year. Those intensified tides could have influenced coastal habitats, perhaps creating more extensive tidal pools and intertidal zones, places ripe for evolutionary experimentation, as life adapted to periodic exposure and changing salinity. Creatures like Dickinsonia, often described as a flattened, ribbed oval, became icons of the Ediacaran because of their unusual symmetry and repeated segments. Sprigina hints at a head-like end and suggests directional movement. Charnia resembles a frond or feather gently waving in a current, anchored at its base. Tribrachidium looks like an elegant three-armed star, its symmetry unlike the bilateral symmetry most animals later adopted. Many of these forms defy easy classification, and that defiance is part of their beauty. They invite us to look beyond our categories and imagine body plans that evolved on a planet with different ecological rules. Not all Ediacaran life was soft and mysterious. By the very end of the period, the fossil record begins to show mineralized, hard-bodied organisms with shells or protective structures. These early experiments with biomineralization would set the stage for the rich fossil record of the Cambrian, when hardened shells and skeletons fossilize far more readily and leave a clearer trail of evolutionary history. Visiting an Ediacaran seashore in the imagination is to step into a pale painting. Low, broad waves slapping a coastline, tides pulling away to reveal mats of bacterial film, anchored fronds swaying in the shallows, and quilted bilaterally repeated forms slowly exploring the substratum. It's quieter than a modern reef in terms of visible motion, but not empty. Instead, it is full of subtle traffic, slow creeping, filtering, anchoring, and the slow compositional choreography of ecosystems finding ways to exploit light, chemical gradients, and shelter. Which brings us to the human angle. These discoveries remind us that life's history is not a linear ladder leading inevitably to us. Instead, it is a branching, experimental process filled with lost avenues and once successful designs. The Ediacaran Biota includes lineages that paved a route toward familiar animal groups, but it also includes unique lineages that went extinct, leaving fossils as their sole testament. Their strange forms expand our imagination of what a living creature can be. Finally, the Ediacaran is a lesson in patience, persistence, and curiosity. It took decades of globetrotting fieldwork, painstaking geochemical analysis, and revisions of classification to reveal this chapter. Its study shows how geology and biology intertwine. Rocks hold chemical signatures of planetary change. Fossils record life's responses. Together, they tell a story of a planet emerging from deep frost into a world of new possibilities, where life quietly, then more boldly, shaped itself into ever more complex arrangements. As the narrator, I leave you with one lasting image. A shallow sea with a carpet of life unlike anything we see today, a mosaic of fronds and quilts and discs, each an experiment in being big, multicellular, and different. They lived by ancient currents and under a brighter, nearer moon. And when their world changed, through evolving predators, shifting chemistry, or climatic swings, their impressions remained, fossilized in rock, waiting for a future mind to read them. The Ediacaran is that fossilized whisper, an invitation to imagine other ways life can organize itself, and a reminder that Earth's history is rich with stories that continue to teach and astonish. Ambient synth fades into a gentle chime as if closing a chapter. The narrator's voice softens. Consider the Ediacaran not as an alien past disconnected from us, but as the slow rehearsal for life's great diversification, a series of first steps, missteps, and daring experiments that set the stage for the animal world we recognize. Its fossils are both relics and lessons, relics of forms that once were, and lessons about the boundless inventiveness of life when a planet gives it the space to try something new. The final note lingers, leaving room for wonder.